In this second part of a review, we are going to begin with carbohydrates. Carbohydrates are molecules made of carbon and water. The monosaccharides are made of only one unit of carbohydrates, and the three main examples are glucose, galactose, and fructose. Remember that you have to recognize the formulas of these three compounds. It's also important that you recognize which one is D-glucose from L-glucose according with the position of the OH in the chiral carbon. Remember that these compounds can be represented like the Fischer projection, but in the real life they are in form of cycles, as you can see here. According with the position of the OH in the chiral carbon, if it is down, it is alpha D-glucose, for example. If it were up, it would be beta D-glucose. The disaccharides that we studied were maltose that were made of 2 units of glucose, sucrose that is made of glucose and fructose, and lactose that is made of glucose and galactose. Remember that the bond between 2 units of monosaccharides is known as glycosidic bond. The polysaccharides are compounds that are made of more than 1 unit of glucose. In the case that we can see on this slide are the ones that belong to the plants and the one that belongs to the animal. Glycogen is in the animals, while the starches, amylose and amylopectin, and cellulose are in the plants. Cellulose is part of the structure of the plants, while amylose and amylopectin are a form of storage in plants, while glycogen is a form that we store glucose in our bodies. The cellulose is linear, but between the different strands we have hydrogen bonds. That's why cellulose is so hard. And amylose is also linear, but the bond is alpha 1,4, while in glucose is beta 1,4. Both are linear, but the bonds are different. Amylose and amylopectin and glycogen are both branched, but as you can see here, glycogen is more branched than amylopectin. Both contains 1,4 and 1,6 alpha bonds. The fatty acids, saturated and unsaturated, are 2 units that correspond to the lipids. The union of 3 fatty acids to a glycerol skeleton is what we call a triglyceride. Triglycerides are dangerous if the level is so high, because can produce, as you can see in this figure, plaques together with the LDL cholesterol, and it can produce heart attacks. In the case of the phospholipid, instead of having 3 units of fatty acid, we have a phosphate group in one of the carbon of the glycerol. Phospholipids are very, very important because they made the bilayer of the cell membrane, as you can see in this figure. Another important lipid is the arachidonic acid that is in all the cells in our body. The mechanism of conversion of the arachidonic acid into the corresponding prostaglandin produces pain, produces bleeding, they produce inflammation or fever. If we break down this mechanism by using enzymes, we stop having fever, pain, or whatever. The other group of lipids that we study were the steroidal. The main compound is cholesterol. How can we recognize steroids? Because of these three hexagons joined by this pentagon. This is the skeleton of all the steroidal compounds. Cholesterol could be HDL, that is the good one, and LDL, that is the bad one, because together with triglycerides produce plaques that lower or doesn't allow the blood to go through it and can produce heart attacks. But cholesterol is also important because it's a raw material for sexual hormones. For example, here we see in the menstrual cycle how in the first step, in the first 14 days, the progesterone is higher because it allows the production of the ovulation, while in the second part, if the ovule is fertilized, then progesterone lower the level, but while estrogen is secreted more in order to produce to make the possibility here I said first estrogen is the one that makes possible the ovulation, while progesterone is the one that makes possible the preparation of the uterus walls to receive the egg fertilized. In men, the level of testosterone makes different changes in the bodies. As you can see here, according with the age, the production of testosterone lowers. Actually, many people are using testosterone to produce muscle, but it has effects that are not good for health. The amino acids are those compounds that are made of an amine group and a carboxylic acid group. The one that makes the difference is this side chain that makes the different amino acids, as you can see here, with different characteristics according with this group, that can be non-polar, charged, polar, and the join of one amino acid with another amino acid by a peptide bond produce a dipeptide. If we have more than 100 amino acids, we have a protein. Proteins are compounds that in the original or primary structure are strands joined by peptide bonds. But it can have a secondary structure, that is when you have two strands joined by hydrogen bonds, and here we have the alpha helix and the beta pleated sheet. In the case of the tertiary ones, we have several interactions like hydrophilic interaction, salt bridges, disulfide bonds, and so on. The union of several tertiary structure produce the globular structure in the proteins, or quaternary structure. Here we can see how different, according with the structure of the protein, they can have different functions. regulation, structural movement, and so on. Let's continue by an example of the human insulin. As you can see here, it's very important because it regulates the level of glucose in blood. Also, another important function is enzymes. The enzymes have an active site where the substrate is binded and the enzyme produces the bond that it joins the units, the different units in the substrates, is broken and release these simple units, while the enzyme is ready to begin working once again. The DNA and RNA are the nucleic acids. The building block of nucleic acids are the nucleotides that are made of a phosphate group and a sugar and the nitrogen base. The nitrogen bases can be in RNA, cytosine, guanine, adenine, and uracil, while in DNA are cytosine, guanine, adenine, and thiamine. The difference here is uracil per thiamine. RNA is single strand while DNA is double strand. The sugar in RNA is ribose while the sugar in DNA is deoxyribose. It's very important for you to study what is the replication and what is the transcription. In the replication, the DNA reproduces itself by joining different nucleotides, and this process is semi-conservative because an old strand joins to a new one, and then we have two new ones. In the case of transcription, the process is to copy the information of DNA by an RNA, in this case messenger, that is the one that takes this information to the cytoplasm, in specific to the ribosome, to produce the proteins by a process that is known as translation. Different nucleotides carry the different amino acids that join together to produce the proteins. As you can see, proteins are everything in our body. Well, that's all. Try to go over all this content and you will be fine in the final exam. Good luck.